Hi, this is Dr. Kat Vlies from Central New Mexico Community College. We're going to focus in this video on the neuromuscular junction. In the previous video, I explained to you that a neuromuscular junction is a synapsis between an axonal terminal with its synaptic bulb of a somatic motor neuron and a skeletal muscle cell or a muscle fiber. And so here I'm drawing the sarcolemma, and notice that nearby that axonal terminal, it is very convoluted. This right here illustrates a T-tubule, and perhaps there is another T-tubule over here. That also means that I need to add some of our sarcoplasmic reticulum, and rather than drawing all of the sarcoplasmic reticulum, I'm just going to show you the terminal cisternae as these sac-like structures like so that give rise to this tubular system. And on either side of these T-tubules, remember, we always have a terminal cisterna. TC for terminal cisterna, T-tubule right here, okay. Here's our axonal terminal, and inside of these axonal terminals, or synaptic bulbs of these axonal terminals, we have little vesicles, and we call these synaptic vesicles. They contain neurotransmitters. And the neurotransmitter that is always going to be released onto skeletal muscle is going to be acetylcholine, always. It will never be any other neurotransmitter. There are many neurotransmitters in our body, especially in our central nervous system, but pretty much throughout our body. And some examples that you're familiar with are norepinephrine, dopamine, serotonin, just to name a few. But the one that always lands on skeletal muscles and that is always being released by the synaptic vesicles of our somatic motor neuron is acetylcholine. So or I should say, if this is our synapses right here, then right here, this cleft, this little bit of a space, we express it in nanometers, so it's very minute. We refer to that as a synaptic. That's the synaptic cleft. And it is just filled with the material that you find around any cell, namely extracellular material. Extracellular material in the synaptic cleft around our cells, and therefore also inside of the T-tubules. So your T-tubules are filled with extracellular material. It's just as if that watery material continues to flow into our T-tubule. Let me grab a better pen to illustrate these T-tubules because they're not showing up very well. So here we go. They're just invaginations of our sarcolemma. And then finally, let's not forget that we have our thick and thin filaments inside of our sarcoplasm organized inside of myofibrils. Again, I'm not going to draw that very detailed, but this is going to represent those myofibrils with their thick and thin filaments. So the idea is for all of that calcium that is stored in a high concentration inside of the terminal cisternae is for that calcium to be released and then make it to the thin filaments where our troponin complex is located. Bind with the troponin complex, change its three-dimensional shape, and that will allow for our thick filaments to bind. But how does it all work? Well, from the central nervous system, either the brain or the spinal cord, an electrical signal arises, and we call that an action potential. And that action potential will come down all the way down the axon towards all of the axonal terminals of the axon. And here we see one axonal terminal. And so that action potential, which I'll illustrate with an arrow here, arrives. Well, this action potential consists of the moving around, the flowing around of many ions. It's basically a current. And that is going to disturb the resting membrane potential of our axonal terminal. Every cell in your body is characterized by this gradient of electrical charges across the cell membrane, which we call the resting membrane potential. And only in neurons and in muscle cells, any muscle cells, can that resting membrane potential be disturbed. And when it's disturbed, it results from the opening of various ion channels. So we need to talk ion channels, and we're going to therefore also have to specify ion concentrations. We've already mentioned that the ion concentration of calcium inside the terminal cisternae inside of our muscle cell is very high. Very high compared to what? Compared to the concentration of calcium inside of the sarcoplasm. So I'm trying to write it a bit smaller here to imply that the concentration here is less. But we can't just focus on calcium here. Calcium also plays a role at the level of the axonal terminal. And inside of our axonal terminal, we see that the calcium concentration is lower than outside. So I'm going to write a big calcium symbol right here. I hope you can see that. Let's see if we can make that stand out a little bit better with blue, like so. And so outside any cell, whether it's our neuron or our muscle cell, we're going to see that the calcium concentration is going to be much higher than inside. So our calcium concentration outside our muscle cell is much higher than in its sarcoplasm. But if we compare the concentration of calcium in the terminal cisternae with that of the sarcoplasm, the terminal cisternae is much higher. And remember, ions always follow their concentration gradient. We have to add two more ions. One is potassium. Potassium has, in all of our cells, it doesn't matter which cell, has a much higher concentration inside of cells than on the outside of cells. And the opposite is the case for sodium. So inside of cells, we see a much lower concentration of sodium compared to the outside. And so maybe we can add another sodium here. High concentration there, much smaller here. And the opposite for potassium, much higher on the inside of a cell compared to the outside of a cell. So if there's something that could trigger